So we're going to take a look at the regulation of the heart. The heart has to adjust to demands that change over time between a resting state and a more active state, particularly the exercising state. And during exercise, for example, the heart beats faster and pumps more blood out of it than at rest. So there's a term known as the cardiac output that is important to understanding the regulation of the heart. And the cardiac output is the amount of blood pumped out by each ventricle per minute. And the cardiac output is determined by multiplying the heart rate times stroke volume. So the heart rate is the number of beats per minute, and even though it's a range, it's somewhere around 75 beats per minute. The stroke volume is the amount of blood pumped out of each ventricle each time the heart beats one time. And it's approximately, in a resting individual, 70 mils per beat. So if we calculated the resting cardiac output, it would be 75 beats per minute, that's the heart rate, times 70 mils per beat, the stroke volume, and we can cross beats as units because it appears above and below the divisor. So we end up with milliliters per minute, and it's around 5,250 milliliters per minute. So there's 1,000 milliliters in a liter, and this would be the same as 5.25 liters per minute. That's the resting cardiac output. The output of the heart actually increases dramatically, fourfold, during exercise. So the exercising cardiac output might have a heart rate of 150 beats per minute or more, times a stroke volume of 140 mils per beat, because the heart is beating more forcefully and pumping more blood out, and that would equate to 21 liters of blood moving through the heart each minute during exercise. We're going to look at the regulation of stroke volume. So the heart in a resting individual only pumps out about 50, maybe 60% of the blood that flows into it. So it's not pumping all the blood that flows into it out of the heart. During exercise, when the ventricles contract more forcefully, the heart will pump out as much as 90% of the blood coming into it with each beat. So stroke volume can change between a resting and an exercising state. There's a couple terms here that I want you to know that we need to know in order to calculate stroke volume, and that would be the end diastolic volume and the end systolic volume. So the EDV, or end diastolic volume, is the volume of blood in the ventricles at the end of diastole after they have completely filled. And the end systolic volume is the volume of blood that's left in heart chambers after they've pumped blood out during systole. So the stroke volume in a resting individual would be the end diastolic volume, how much blood fills up the ventricles during the resting or diastolic state, and subtract away how much blood is left after systole, and that would be the stroke volume. So if a heart chamber fills up with 120 mils and there are 50 mils left at the end of systole, then it must have pumped out 70 mils of blood, and that would be the stroke volume. How much blood the heart pumps out with each beat, or the stroke volume, is related to what's known as contractility. And contractility refers to the strength of the cardiac muscle contraction during systole. So if we increase contractility, then we increase the strength of the myocardial contractions. The cardiac muscle cells contract more forcefully, and that's going to pump more blood out of the ventricles. In other words, it will increase stroke volume. If we decrease contractility, then the strength of the cardiac muscle contraction weakens. It's not as strong, and that would pump less blood out or decrease the stroke volume. So we're going to find that the sympathetic division of the autonomic nervous system, made up of sympathetic motor neurons releasing norepinephrine as a neurotransmitter and epinephrine from the adrenal gland, will increase contractility and increase stroke volume. So sympathetic neurons act to increase contractility and increase stroke volume. On the other hand, parasympathetic motor neurons secreting acetylcholine will decrease contractility and as a result decrease stroke volume. So there's a law of the heart that is kind of interesting called the Frank-Starling law of the heart. It almost looks like it's named after somebody called Frank Starling, but there was two individuals, one with the last name Frank and the other with the last name Starling. And as a result, it's called the Frank-Starling law of the heart because both contributed to its discovery. So the Frank-Starling law of the heart says that increases in the amount of blood flowing through the heart will cause the cardiac muscle cells to stretch, and when you stretch cardiac muscle cells in response, they'll contract more forcefully. And as a result of contracting more forcefully, you would increase stroke volume. So cardiac muscle cells are said to act like rubber bands. The more they stretch, the more forcefully they contract or recoil back to their original length. So you take a rubber band like this and you stretch a little bit and you get this as the force of recoil. But if you stretch a whole lot more, the recoil force, or the force of contraction in this case, would be a lot stronger. So this law, the Frank-Starling law of the heart, is important during exercise because it means the heart can self-regulate stroke volume to a certain extent. During exercise, more blood flows through the heart, the cardiac muscle cells stretch as a result and contract more forcefully to increase stroke volume. So before I go any further, I just wanted to do a quick review of the nervous system. So the nervous system consists of a central nervous system and a peripheral nervous system. The central nervous system would be the brain and spinal cord. The peripheral nervous system, or PNS, would be everything outside of the brain and spinal cord. So it's going to include the cranial and the spinal nerves and all of their branches throughout the body, as well as sensory neurons and the receptors that pick up sensation of pain, touch, temperature change, smell, and motor neurons. Motor neurons, in turn, are divided into somatic motor neurons and autonomic motor neurons. And the autonomic motor neurons are further divided into parasympathetic or sympathetic neurons. So there's a couple types of motor neurons: somatic motor neurons and autonomic motor neurons. Somatic motor neurons are motor neurons that stimulate skeletal muscle cells to contract. Somatic motor neurons release a neurotransmitter called acetylcholine, and when acetylcholine is the neurotransmitter, the effects on the target cells, in this case skeletal muscle cells, are called cholinergic effects. Somatic motor neurons are said to be under voluntary or conscious control. In other words, you have to will them into action. You have to say, hey, bicep muscle contract, and then it will contract. And you have to be conscious in order to control the skeletal muscle. So when you're sleeping, you're in an unconscious state, and you wouldn't expect to, have the skeletal muscles contracting. Autonomic motor neurons, on the other hand, are motor neurons that are automatically active, and they're under unconscious or involuntary control. You don't have to think about stimulating autonomic motor effects. Autonomic motor neurons affect cardiac muscle cells, smooth muscle cells, and glandular epithelial cells. There are two divisions of the autonomic nervous system, the parasympathetic division and the sympathetic division. And we'll talk a little bit about each. The parasympathetic division of the autonomic nervous system is made up of parasympathetic motor neurons that release the neurotransmitter acetylcholine, hence they have cholinergic effects. And the parasympathetic effects promote or encourage resting and digestive functions. So the parasympathetic effects on the heart include a decrease in heart rate. And that helps to maintain a resting heart rate. The sympathetic effects on the body are mediated by sympathetic motor neurons that release a neurotransmitter called norepinephrine, which I'll abbreviate NEPI. And a sympathetic response also results in the release of epinephrine, a neurohormone from the adrenal gland. And when norepi and epi are having their effects on target cells as a result of a sympathetic response, those effects are called adrenergic effects. The sympathetic neurons are activated when the body is stressed. And there's two major categories of stressors. You have psychological stressors, fear, anxiety, and guilt. Anxiety is a little different than fear. Anxiety is the fear of the unknown. So you might have test anxiety. In other words, you are anxious or a little fearful as to what your grade might be, and that's why you are anxious. Physiological stress is stress like exercise or low blood sugar levels, hypothermia, where the core temperature of the body has decreased to dangerously low levels. And the sympathetic adrenergic effects on the cardiovascular system include an increase in heart rate, an increase in stroke volume, as well as an increase in cardiac output and an increase in the blood pressure. So we're going to take a look at the regulation of the heart by the autonomic nervous system. And as we do, we'll talk about parasympathetic and sympathetic effects. So there are four major regions of the brain. You have the cerebrum, diencephalon, cerebellum, and the brainstem. The brainstem is the portion of the brain that gives rise to the spinal cord. And we're going to take a look at autonomic centers found in the medulla oblongata of the brainstem. And there's three of them. The cardioinhibitory center, which I'll abbreviate CIC. It's a parasympathetic center that acts to decrease heart rate. The cardioaccelerator center, or CAC, which is a sympathetic center that acts to increase the heart rate and stroke volume. And the vasomotor center that we'll talk about a little bit later, which is a sympathetic center that acts to increase what's known as the peripheral resistance, which in turn will increase blood pressure. And all three of these autonomic centers are located in the medulla oblongata of the brainstem, one of the major regions of the brain. So before we look at some of the details, let's just have an overview, because that's really what you want to be able to see, the big picture. And we'll find that the CIC, parasympathetic center, through the release of acetylcholine, will decrease heart rate. The other two centers are sympathetic centers, the CAC and the vasomotor center. The CAC, or cardioaccelerator center, through norepinephrine and epi, these will be adrenergic effects, will increase heart rate and stroke volume. And we're going to find that the vasomotor center is associated with what's called the peripheral resistance and blood pressure. So the cardioinhibitory center is a group of parasympathetic motor neurons found in the medulla oblongata of the brainstem. And axons of these parasympathetic neurons travel out of the brainstem by way of the vagus nerves. They'd be cranial nerve number 10, and travel off in a branch off the vagus nerve to the heart. The parasympathetic neurons release a neurotransmitter called acetylcholine and have what are called cholinergic effects on their target cells. In this case, the target cells will be those of the heart. The acetylcholine effects on the heart include the following. Acetylcholine from parasympathetic motor neurons will decrease the SA node firing rate to decrease heart rate. And acetylcholine will increase the AV node delay time. And that means it takes the cardiac impulse longer to spread across the heart, and that contributes to a decrease in heart rate as well. So this just shows a picture of the cardioinhibitory center. Up here would be the brainstem, and this would be the medulla oblongata portion of the brainstem. It's the medulla oblongata of the brainstem that continues as the spinal cord. Within the medulla oblongata, we find the cardioinhibitory center. Half of it is on the right, half on the left. Both halves together make up one cardioinhibitory center. The cardioinhibitory center contains parasympathetic neurons, and the axons of these neurons travel out the vagus nerves and then out a branch off the vagus nerves to the heart. And parasympathetic neurons will decrease the SA node firing rate and increase the AV node delay time, and both of those effects act to decrease the heart rate. The cardioaccelerator center is a group of interneurons found in the medulla oblongata of the brainstem, and the axons of these interneurons then travel down the spinal cord to stimulate sympathetic motor neurons in the spinal cord gray matter. The axons of the sympathetic motor neurons go out of the spinal cord and travel to the heart as well as the adrenal gland. Sympathetic neurons to the heart release norepinephrine as the neurotransmitter, hence the effects are adrenergic. And sympathetic neurons also go to the adrenal glands and stimulate the release of epinephrine into the blood, and then epinephrine travels by way of the blood to the heart. Some of the adrenergic effects of norepinephrine and epinephrine include an increase in the SA node firing rate, which will increase heart rate, a decrease in the AV node delay time, so the impulse spreads across the heart more quickly, and that contributes to an increase in heart rate. And adrenergic compounds, norepi and epi, will increase the contractility of cardiac muscle cells. So they contract more forcefully, and that acts to pump more blood out of the heart or increase stroke volume. This is a schematic showing the cardioaccelerator center. So here's the medulla oblongata of the brainstem that gives rise to the spinal cord. And within the medulla oblongata of the brainstem, next to the CIC, which I'm not showing, would be the two halves of the cardioaccelerator center. The cardioaccelerator center consists of interneurons that leave the CAC and travel down into the spinal cord, where they stimulate sympathetic neurons that travel to the heart, and sympathetic neurons that travel to both adrenal glands. The adrenal glands are above the kidney. And sympathetic neurons to the adrenal glands stimulate cells in the adrenal gland to release epinephrine into the blood, and then epinephrine travels by way of the blood to the heart. So norepi from sympathetic neurons that go to the heart increase the SA node firing rate, decrease the AV node delay time, and increase the contractility of the ventricular myocardium. All of these effects together dramatically increase heart rate and increase stroke volume. Epinephrine has the same effects as norepi. Norepi from sympathetic neurons is a neurotransmitter, and epi, because it's released into the blood and travels by way of the blood to the heart, is considered to be a neurohormone. So there's a couple heart rhythms that I want you to know, and one is simply the normal resting heart rate, which is somewhere around 70 to 75 beats per minute. The range of normal would be anywhere from 60 to 100 beats per minute. So we're going to take a look at several arrhythmias, and an arrhythmia is some kind of abnormal beat pattern to the heart, where the beat pattern of the heart is the rhythm of the heartbeat. So tachycardia is an abnormally fast resting heart rate that would be over 100 beats per minute or outside of the normal range. And bradycardia, where brady means slow, is an arrhythmia where you have an abnormally slow resting heart rate. That would be below 60 beats per minute. A myocardial infarction, or an MI, is more commonly referred to as simply a heart attack. So just a little dissection of myocardial infarction. The myocardium would refer to the cardiac muscle cells in the middle layer of the heart. An infarct is when a group of cardiac muscle cells die due to what's called ischemia. And ischemia is when you have a lack of oxygen and nutrients to cells because of a decrease in the flow of blood. The most common cause of a heart attack or a myocardial infarction is coronary artery disease, CAD. And coronary artery disease is caused by atherosclerotic plaque that builds up in the walls of coronary arteries. The coronary arteries are the major arteries that branch off the aorta that deliver oxygen and nutrients throughout the heart. So a thrombus is an abnormal clot that can form on the sides of the atherosclerotic plaque. And if a piece of the plaque breaks off and drifts away in the blood, that piece of plaque that broke off is known as an embolus. The embolus can get stuck as it drifts in a small artery where it can't go any further, and that would block the flow of blood through that artery to cardiac muscle cells downstream. Those cells, as a result, would suffer from ischemia, the decrease in blood flow and a decrease in oxygen and nutrients, and may die as a result. If they do, you suffer a heart attack or an MI. The dead and dying cells associated with the MI generally cause some chest pain, shortness of breath, and dizziness. And an MI can lead to what's called ventricular fibrillation. And that's cardiac arrest, and that can lead to death. So let's talk a little bit more about ventricular fibrillation. Ventricular fibrillation, like tachycardia and bradycardia, would be classified as an arrhythmia. And ventricular fibrillation is also known as cardiac arrest. So ventricular fibrillation is an arrhythmia where honestly the heart stops beating. And as a result, it doesn't pump blood to the organs of the body, to include your brain. So if the heart stays in ventricular fibrillation, where it's not beating and pumping blood out, for 4 or 5 minutes, you aren't delivering oxygen and nutrients to the cells of the body, but because you don't have oxygen and nutrients going to the brain, which is a vital organ, brain cells can start to die. And if you don't somehow correct ventricular fibrillation, it would result in massive damage that's irreversible to the brain and death. So there is a procedure called a defibrillation. And defibrillation is often done by what's called an external defibrillator. And that's a device that you place on an individual's chest and deliver a really strong electric shock to their chest and from there to their heart with 700, 800, 1,000 volts. And that, hopefully, the electric shock will cause all the cardiac muscle cells to stop any kind of activity. And the hope is that the SA nodal cells will fire and then start to control the heartbeat and return it to normal. We're going to look next at the control of blood pressure. So blood pressure is the force that blood exerts against the sides of blood vessels. We've seen that resting systemic arterial blood pressure is somewhere around 120/80, and the resting pulmonary arterial pressure, which is a much lower pressure system, is somewhere around 25/10. So when one talks about blood pressure, they're almost always talking about the systemic arterial blood pressure, and that'll be true in this section. If someone's talking about the pulmonary pressures, they're obligated to let you know that. So blood pressure, mathematically, is the cardiac output times peripheral resistance, and because cardiac output equals heart rate times stroke volume, we can expand this equation and say that blood pressure is equal to heart rate times stroke volume times peripheral resistance. So when you increase the heart rate or the stroke volume or the peripheral resistance, multiplicatively, you would increase blood pressure. So peripheral resistance, or PR, is the resistance that blood vessels give to the free flow of blood. The peripheral resistance is directly related to friction, and friction is something that occurs when two objects rub against each other. So the red blood cells in blood rub up against the sides of blood vessels, and there's trillions of them circulating in the blood at any given time. And as red cells rub against the sides of blood vessels, they create friction that slows down the flow of blood. We're going to find that the diameter of blood vessels is inversely proportional to the peripheral resistance. An inverse or reciprocal relationship means that two variables are always moving in opposite directions. So if one variable goes up, the other is going down, and vice versa. So we're going to find that diameter is inversely proportional to the peripheral resistance. So if we increase the diameter of a blood vessel, its peripheral resistance will drop. And if we decrease the diameter of a blood vessel, its peripheral resistance will go up. Vasomotion. We'll find that peripheral resistance is controlled by something called vasomotion. And vasomotion is what causes changes in the diameter of arteries and arterioles. So there's two types of vasomotion. One is called vasoconstriction, and it increases peripheral resistance. Vasoconstriction occurs when the smooth muscle cells in the walls of a blood vessel contract, and that decreases the diameter of the blood vessel and acts to increase the peripheral resistance. So if we started with a blood vessel like this, and you look at the diameter when the smooth muscle in the walls contract, the blood vessel diameter gets smaller and smaller and smaller, and that's vasoconstriction. On the other hand, vasodilation, which decreases the peripheral resistance, is when the smooth muscle in the walls of blood vessels relax, and that causes the diameter of the blood vessel to increase, which would decrease the peripheral resistance. So when the smooth muscle in the walls of a blood vessel relax, then the diameter of the blood vessel increases, and as it does, the peripheral resistance would decline. So let's look at some of the autonomic nervous system effects on blood pressure. The CIC, which is the parasympathetic center, acts to decrease heart rate, and a decrease in heart rate leads to a decrease in blood pressure. The CAC, which is a sympathetic center, acts to increase the heart rate and increase stroke volume, and that in turn would increase blood pressure. The vasomotor center is a sympathetic center found in the medulla oblongata of the brainstem, along with the CIC and the CAC. And the vasomotor center leads to a stimulation in sympathetic neurons that cause the vasoconstriction of arteries and arterioles, and that decreases the diameter of these blood vessels, particularly the arterioles, which leads to an increase in peripheral resistance, and if we increase peripheral resistance, we'll increase blood pressure. There's an interesting reflex that I want you to know about. It's a homeostatic reflex called the baroreceptor reflex. And it's a mechanism that helps to regulate blood pressure. So it involves stretch receptors that are found in the walls of the aortic arch and the carotid arteries. And these stretch receptors are baroreceptors. And they monitor changes in blood pressure. So here's a normal blood pressure. And let's assume the blood pressure goes up. That'll stimulate the baroreceptors, and the baroreceptors then will send sensory impulses into the medulla oblongata of the brainstem, where these sensory impulses from baroreceptors, stimulated by an increase in blood pressure, will decrease the sympathetic centers, the CIC and the vasomotor center, and increase the parasympathetic CIC. And the net result then is to cause a decrease in heart rate, stroke volume, and peripheral resistance, and the net effect then is to bring the blood pressure back to normal. You've also got chemoreceptors that help to regulate blood pressure. They help to regulate the breathing rate and depth as well. So chemoreceptors are found in the walls of the aorta and the carotid arteries. They're also found on the sides of the fourth ventricle of the brain. The chemoreceptors monitor changes in the concentration of carbon dioxide in the blood, as well as hydrogen ions and oxygen. Definitely remember that the chemoreceptors are most sensitive to CO2 changes. In other words, a small change in the CO2 concentration of the blood will stimulate these chemoreceptors, where it would take a much larger change in the oxygen levels to have the same effect. So there's a reaction that you need to commit to memory. It comes up in a few of the topics that we talk about this semester. We'll come back to it during the respiratory system and the urinary system, and we'll find that you just need to know this reaction. So carbon dioxide, which is the gaseous waste product produced by cells as they make ATP by cellular respiration, carbon dioxide will react with water to form an acid called carbonic acid. And carbonic acid then will split apart into hydrogen ions and bicarbonate ions. So if the CO2 levels go up in the body, it will drive this reaction to the right and generate more and more hydrogen ions, which will cause a drop in the pH and a tendency towards what's called acidosis. Acidosis is lethal. and it will kill cells and ultimately kill the individual. So you want to prevent carbon dioxide from accumulating in the blood and driving this reaction towards the state of acidosis. And of course we do that by exhaling the CO2 as it forms out of our body. The carbon dioxide that's generated as a result of cellular respiration will trend the body towards acidosis. So the lungs have to get rid of the CO2 so it doesn't accumulate in the body. So this is kind of a flow chart for how the chemoreceptors engage to help regulate CO2 levels in the blood. We start with a normal amount of CO2, and let's assume that an individual goes out and exercises. That will step up the metabolism of skeletal muscle, and skeletal muscle as a result would generate more ATP and carbon dioxide as well. So the carbon dioxide will eventually react with water, and that leads to the formation of an acid that increases the hydrogen ion concentration and can lead to acidosis. So you have to get rid of this extra CO2 that would be formed during exercise. The high levels of CO2 that develop in the blood will stimulate chemoreceptors in the aorta, the carotids, and the fourth ventricle of the brain. And that will cause two things to occur. One of the effects of chemoreceptors is to stimulate an increase in the rate and depth of breathing, and as a result you would exhale more CO2 out of the body to help bring it back to normal. The other effect of the chemoreceptors is to send impulses into the medulla oblongata of the brainstem, where the chemoreceptors will stimulate sympathetic centers, the CAC and the vasomotor center, while inhibiting the activity of the cardioinhibitory center. The net effect is to increase heart rate, stroke volume, and peripheral resistance, all of which taken together will increase the pressure of blood, and that in turn will move more blood to the lungs to get rid of that carbon dioxide. I want to just mention autoregulation of blood flow through exercising skeletal muscle. So autoregulation means that skeletal muscles can actually regulate the amount of blood that flows through them. And here's how that works. So let's assume normal blood flow through resting skeletal muscle, and then one goes out and exercise. So with exercise, you're going to increase the activity of the skeletal muscle, and that's going to increase the need for ATP, and cellular respiration provides that ATP by breaking down organic compounds, and of course we're going to generate carbon dioxide. So the increase in carbon dioxide and an acid called lactic acid in the fluids around the skeletal muscle cells triggers two things to occur. One, vasodilation of arterioles in exercising skeletal muscle, as well as the relaxation of smooth muscle, rings of smooth muscle or bands of smooth muscle called precapillary sphincter muscles. And both of these effects increase blood flow through the exercising skeletal muscle and the flow of blood to deliver oxygen and nutrients to the skeletal muscle cells through capillaries. The net effect of increasing blood flow into capillaries means that you're going to deliver more oxygen and nutrients to the exercising skeletal muscle and pick up the additional CO2 to flush or drain it out of the exercising skeletal muscle. And this ability to autoregulate is called autoregulation, and it occurs in exercising skeletal muscle. A little bit about blood vessels. So the walls of arteries, arterioles, as well as venules and veins is three-layered. The outer layer is called the tunica externa. The layer in the middle is the tunica media, And that's the thickest layer, where you'd find mostly smooth muscle cells as well as elastic fibers that help to increase the stretchability or the elastic nature of the blood vessel wall. Traveling throughout the tunica media would be the axons of sympathetic motor neurons involved with controlling vasomotion. The innermost layer is the tunica intima, and it's primarily simple squamous epithelium or endothelium, and it would line blood vessels. The arteries are the blood vessels with the thickest tunica media, and it's thick because they have to withstand the highest blood pressures that develop. Arterioles are very small, with diameters less than 1/10 of a millimeter, and they're important in regulating blood pressure through vasomotion. Metarterioles and thoroughfare channels. So metarterioles are short blood vessels that link arterioles to capillary beds, and thoroughfare channels are blood vessels that drain blood from capillary beds into venules. So this would be an arteriole all the way down, and arterioles don't just feed one capillary. Instead, arterioles can feed several capillary beds or capillary networks. So this is a metarteriole. Here's another metarteriole and another metarteriole branching off the arteriole, and the metarterioles link the arteriole to a network of capillaries. So where there's one capillary, there would be somewhere between 10 to 100 all interconnected and forming what's known as a capillary network or a capillary bed. The capillary networks are drained by thoroughfare channels that then deliver the blood coming out of the capillary network into a venule. So metarterioles into capillary beds that are drained by thoroughfare channels into a venule. But do note that one arteriole can feed blood into a number of capillary networks. Capillaries are microscopic, and that means that you can't see them with the naked eye. The wall of a capillary is endothelium, in other words, simple squamous epithelium, so it's very thin. And capillaries form capillary networks, also called capillary beds, that consist of 10 to 100 capillaries that are all interconnected with one another. Capillaries are permeable, and the word permeable comes from permeate, which means to move through. They're the only blood vessels that are permeable. Any kind of exchange that occurs always occurs across capillary walls. The arteries, arterioles, venules, and veins are not permeable, and there's no exchange across them. So we're going to find that certain chemicals, and of course white blood cells as well, can move across capillary walls. So there are pores or openings in a capillary wall, actually openings through the endothelial cells, and there are openings between cells. A precapillary sphincter is a circular arrangement of smooth muscle cells at the entrance to a capillary bed, and if the precapillary sphincter is contracted, blood doesn't flow into the capillaries, where if it's relaxed, blood does flow into the capillaries. Venules and veins have the same three-layered construction as arteries and arterioles. So there's a tunica externa, tunica media, and a tunica intima. The walls of venules and veins are thinner than arteries and arterioles. The tunica media in veins is thinner because there's simply less smooth muscle within it, because veins don't have to withstand the same kind of pressures that we see in the arteries. The veins contain one-way valves, and these are cup-like structures on the side of the vein that only allow blood to flow in one direction, and that's always towards the heart. So this would be a vein, and these would be cup-like structures on the sides of a vein at regular intervals. And the blood flows from one valve section to the next, and then from this valve section to the next valve section, always moving towards the heart. So if the blood attempts to go in a reverse or retrograde fashion, it will fill up these cup-like flaps, and they will swell outwards to seal off this opening right here, which wouldn't allow the blood to flow backwards. The venous system is a low pressure system, so there are a couple factors that affect the return of blood in the venous system. The blood pressure in the systemic veins is probably around 15 millimeters of mercury in the venules, and it'll drop below 5 millimeters of mercury in the major veins, like the inferior vena cava. As a result, it's very difficult to move blood effectively in systemic veins back to the heart because it's such a low pressure system and the blood moves rather slowly. In addition to the heart, there are two other pumps that help to move blood. So the heart pump is the major pump that imparts kinetic energy to blood, whether it's in arteries or arterioles or capillaries, venules, or veins. The heart is the major pump that creates pressure in blood vessels, all blood vessels. In addition to the heart pump, we've got a skeletal muscle pump and a respiratory pump. So the skeletal muscle pump consists of skeletal muscle that contracts, and when it does, it presses down on veins that run through the skeletal muscle, and that pressure on the veins running through skeletal muscle and between skeletal muscle will cause the blood in the veins to move from one valve section to the next, always towards the heart, because if the blood in veins attempts to move in a retrograde or backward fashion, then the one-way valves prevent that. The other pump is known as the respiratory pump, and it's concerned with the contraction and relaxation of the diaphragm. The contraction and relaxation of the diaphragm helps to move blood in the veins found within the abdominopelvic cavity. And the pump takes advantage of Boyle's law. Boyle's law shows a relationship between air pressure and volume. So the air pressure is inversely or reciprocally related to the volume. An inverse relationship is where two variables are moving in opposite directions. So a decrease in the volume of the abdominal cavity as the diaphragm contracts will increase the air pressure within the abdominal cavity. The increase in air pressure as the diaphragm contracts will press down on the systemic veins in the abdominal pelvic cavity, and that helps to push the blood from one valve section to another, always towards the heart. This is an illustration showing how the respiratory pump works. So the diaphragm is a dome-shaped skeletal muscle at rest. And when the diaphragm contracts, the diaphragm will flatten, and this contraction occurs during inhalation. So the diaphragm is dome-shaped at rest, and then when it contracts during inhalation, it flattens out. And as a result of it flattening out when it contracts, is you decrease the volume by this amount in the abdominopelvic cavity, which increases the air pressure in accordance with Boyle's law. And that increase in air pressure presses down on the blood in veins, causing the blood to move from one valve section to another. Blood can't move in the opposite direction because of the one-way valves.